Hey everyone! Welcome to my final project for Developmental Biology Lab. In this video, I will give an overview of the early Drosophila embryo development. Please forgive any production errors, as this is my first time creating an artistic project of this nature. All the information in this video is available in Developmental Biology 11th edition by Gilbert and Baresi. Any factual errors are my own and are not a reflection of our course material or textbook. Without further ado, let's get started. First up, we have fertilization. In Drosophila, this is unique because the sperm can only enter the egg through one particular spot known as a micropyle. This is to prevent polyspermy, since the Drosophila egg does not have the cortical granules to block polyspermy the way other organisms do. But even though it doesn't have those cortical granules, changes still occur. The sperm enters intact, and the DNA of both the egg and sperm nuclei replicate one more time before fusing. Once fusion occurs, cleavage starts. Drosophila cleavage is characterized by being superficial, cleavage, and syncytial. The earlier means the yolk confines the cleavage to the cytoplasmic rim of the egg, while the latter refers to the fact that chirokinesis occurs without cytokinesis, which creates a syncytium, or in other words, a single cell with multiple nuclei. This happens because the nuclei undergoes several mitotic divisions that alternate between S and M phase without going into gap phase. The cleavage stage has a total of 14 cycles, but I will focus on the two primary ones that meet blast cell transition, of course. Firstly, at cycle 9, 5 nuclei reach the surface of the posterior pole of the embryo. They are then enclosed by the cell membranes and become the pole cells or future adult gametes. Secondly, at cycle 13, the cell membrane folds inward between each of the nuclei and divides each of this energy into a single cell. At this point, the cells are arranged in a single layer around the yolky core, and the embryo now is known as the cellular blastoderm. The cleavage stage comes to an end with the mid-blast cell transition, which takes place between cycle 11th and cycle 14. The MBT stage is where the maternal RNAs are degraded and control is handed to the zygote genes. Shortly after MBT ends, the embryo enters gastrulation. During this stage, the endoderm, mesoderm, and ectoderm are separated. Starts when the would-be mesoderm folds inwardly, which causes the formation of the ventral furrow. This furrow creates a ventral tube in the embryo. Then the endoderm forms two pockets, one at the anterior end and the other at the posterior end. At the posterior end, the pole cells get internalized along with the endoderm, while at the same time, the embryo bends at the anterior end and forms a cephalic furrow. Next, we have the germ band, which is a group of cells along the ventral midline that will give rise to the trunk of the embryo. This germ band forms when the ectodermal cells extend and migrate towards the midline. Once this happens, the body segments start to develop, which divides the mesoderm from the ectoderm. The germ band retracts, placing the segments in the anterior tip of the embryo. At the dorsal end, the epidermal cells migrate and the dorsal closure process takes place. As I mentioned before, during the MBT, the zygote genes take over development. I won't be delving in too deep into the genetic mechanisms for the zygote genes in charge of segmentation and eventual organogenesis. Instead, for sake of brevity, I will summarize the section for you. The first genes to turn on are the gap genes. They are expressed in 3 partially overlapping sections. They, in turn, regulate the transcription of pair rule genes. These genes are activated into 7 bands. These bands lay from dorsal to ventral direction. Each of these bands are made up of 2 parasegments, the posterior part of one segment and the anterior part of the next segment, dividing the embryo into a total of 15 parasegments. They also encode transcription factors that activate the segment polarity genes, which reinforce the boundary of the parasegmentality that was set up by the pair rule genes. They also help determine cell fate through cell interactions in each parasegment. Finally, all three of these genes work together to regulate the homeotic selector genes. These genes specify what structure goes into each of those segments. Next up, we have body axis setup, both anterior, posterior, and ventral dorsal. The anterior posterior axis is set up during oogenesis. For the posterior axis, the nucleus moves to the posterior side of the egg chamber and creates a protein gherkin, which binds to a torpedo, a receptor expressed by the terminal follicle cells. When this happens, the cells differentiate into posterior follicle cells, and at the same time activate protein kinase A in the egg. This protein causes microtubules to orient positive to negative, with the positive end being at the posterior side and the negative at the anterior. Bicode mRNA gets moved to the anterior part of the egg through dynein, and Oscar mRNA gets moved by motor proteins to the posterior part of the egg, where it binds to Nano's mRNA. Vicoid in the anterior will help prevent the translation of caudal, while activating the transcription of hunchback. Both caudal and hunchback are proteins created by the nerve cells and are instrumental in setting up the anterior-posterior body axis. Meanwhile, in the posterior end, nanos will prevent the translation of hunchback. Through the creation of these protein gradients, the anterior-posterior patterning is kept within the embryo. The dorsal-ventral patterning also occurs at this time. It starts with the nucleus traveling on the microtubules to what will become the dorsal side of the embryo, and releasing gherkin, which binds to cells closest to the nucleus. This dorsalizes those cells and prevents the creation of the pipe protein, which then only gets created in the ventral cells. Pipe protein in the ventral cells sulfates the beta-line membrane. These membranes then bind to gastrulation defective, which in turn binds to other proteins and create a cascade that results in ventralizing the cells. And so we come to the end of this video. Thank you for your time and patience. Hope this video was easy to follow and helps anyone that watches it understand the early development of Drosophila. Bye everyone, thanks for watching.